Hi, hello everyone. My name is Veronica Hood. I'm the Chief Scientific Officer for Dravet Syndrome Foundation. It's my pleasure to welcome you all to this webinar today. I see a lot of faces that I recognize as well as new ones, so I'm excited to have a great turnout. Today, we're going to hear about participation in the Dravet Genome Study, which is a study that DSF funded, and it began in 2024. It's the largest project that DSF has ever invested in. We gave the lead researchers Ingo Helbig and Ethan Goldberg and their teams $1 million to sequence the full genome of 500 patients with Dravet syndrome, as well as compare it with electronic medical health records to try to understand more about Dravet syndrome and about the SCN1A gene and background genetics. They've made so much progress partnering with DSF and the community and enrolling a lot of the patients they need, but we still need a few more. And so their team is joining us today to talk to the community more about this project and what they're hoping to accomplish and what we still need to get there. So I am going to hand it over first to Anna Prentice. She's a genetic counselor on their research team, and she's going to kick it off. We have several members of their team on the call, including Sarah Teft, who is a nurse practitioner with the group, and Ingo Helbig, who's one of the, as I mentioned, the leaders of the group. And then I believe we may also have Angela Winters joining. Okay, hi, Angela. Sorry, I missed you join. So we're really excited to have them here to talk to the community in person. Anna, you should be able to screen share if you want. While she's starting to get her screen shared, I see many of you are on camera. That's fantastic. It's also okay if you need to stay off camera. At the end of the presentation, there'll be time for questions, and we can either have those dropped in the chat as you think of them, or there'll be time to ask them straight at the end of the webinar. So just put a question in the chat if you're struggling in the middle, and we'll try to help you out. But otherwise, I'll hand it over to Anna. Thank you.
Veronica. And thank you for the nice introduction. So hopefully we should have everyone here from our team who can answer any questions you could possibly have, because everyone is involved, from kind of the first time that you would email anybody on our team to do the sample collection, to collecting the data, to the data analysis. So any questions you have, we're more than happy to field. But I thought I would start by first talking just a little bit about what this study actually is, why we're doing it, and what we hope to be able to get out of it with your participation. So kind of an overview of this project is that the goal really behind it, the primary goal, is to be able to understand why different people with Dravet syndrome or different people with SCN1A related disorders look different from each other. So we know that, somebody could have the exact same genetic change in SCN1A and have completely different challenges in their life from somebody else with that same genetic change. They might have different types of seizures. Those seizures might, happen earlier in life or later in life, or be harder to control or easier to control. They might have different levels of developmental challenges. One of them might have autism, one of them might not. And beyond knowing that they have a change in SCN1A, we don't really understand why those differences exist. And we want to be able to, because we want to be more specific when somebody is diagnosed with an SCN1A related disorder. We want to be able to say, what are the chances that they'll have those symptoms, more specifically than we can already. So there's kind of two ways that we approach that goal. The first is to, gather together a whole bunch of people who have these genetic changes in SCN1A, including people who have Dravet syndrome, but also people with everything else all along the spectrum, so including GIFs plus, people with gain of function variants who have a really severe early onset epilepsy. We take kind of all comers. And the first step is for us to really understand what symptoms they have and when they have them. So we want to kind of describe their clinical features, and we do that using information that we get from their medical record. So some projects that you might be asked to join can involve, you guys filling out surveys or having to come in for visits, things like that. We don't ask that you do any of that. We just ask that you share the person with the SCN1A related disorder, their medical history with us. So we ask that you give us access to their medical records, and then we do that all on our end of things. So we kind of look at their clinical history and describe it in a more kind of standardized way. And then the second kind of tier of the project is that we will perform genome sequencing for all the people that are enrolled. And a genome sequencing is a genetic test that looks through every single letter of your DNA, letter by letter. It's sort of like a spell checker. And what that does is it tells us what other changes beyond SCN1A that person has in their DNA, whether those changes are rare or common and seen in lots of other people. It tells us all of them. So hopefully, when we have those two things combined together, we can ask the question, for somebody that has worse seizures than somebody else, what other types of genetic changes do they have? Are there ones we see a lot? Are there ones that go along with having these worse symptoms? Or are there genetic changes that we see in people who, maybe had particularly easy to control epilepsy, or whose development was, kind of more on track or more typical than other people's? So we can combine the clinical information from the health records and the genetic information that we get from the genome sequencing to help us understand the differences that we see. What is kind of the output of this project more kind of logistically? So we will have that genome sequencing data, which by itself is a valuable resource. And we'll also have the clinical data that we collect, which is in a format that is really usable by many people. So we're obviously going to use it for our project, but we also want to be able to share some of this data, so the genetic and the clinical information, on a publicly available dashboard. None of that is necessarily identifiable information, so it's not like somebody's name would be shared along with any of that. It's more of a kind of group level data resource, so that people can understand what types of genetic changes people with SCN1A related disorders have, what do their other genes look like, and how do they do clinically over time. And then obviously we also want to have some scientific publications that come out of this study, including one that we're just about ready to kind of submit to journals that's talking mostly just about the clinical data at this point. And then in the future, more publications focusing on the kind of comparison of the genetic data as well. So where are we at right now? Our goal is to have 500 people with SCN1A related disorders enrolled in this project. And right now we have 423 samples. So we still need 77 more participants to hit our goal. And that's kind of why we're here today, is to encourage you guys to help us to hit that goal and to help, kind of push us past the finish line on this project that DSF has so graciously funded. And with that, I will leave it to Sarah Teft, one of the nurse practitioners who works with us, to talk a little bit more about the logistics of how you can participate.
Thanks, Anna. So as we saw on the last slide, we only need 77 more. We really need that amount in samples to be returned. There are a good amount of people we have consented to the project, but we are still waiting for some of their samples to come back. So if you are one of them, this is your kind of reminder also, in that if you see a UPS package laying around, please look inside, because it might be the swab. Feel free to reach out to us if it's been some time. We can help double check if everything is still within like expiration dates and things like that. But it's really easy to sign up. A lot of you might have even just clicked the button to say that to share your information with us, and we'll be following up with you the next week or two. But you can also just email us at scm1a@chop.edu, and we will get back to you and set up a time to do the consent, and then also do the logistical part of shipping things out to you, and then also sometimes getting a release of information so that we can do the background work to get the medical records as well. Um, so once you receive the kit, it's really easy to do the collection. It's just a cheek swab. We have a video in the next slide of how to do it, but we just say, can't eat, drink, chew gum, smoke before the sample's collected. For people who might have rambunctious children, sometimes we'll say to do it kind of like first thing in the morning when they're waking up, just kind of do a little cheek swab. You'll probably go to the video, but basically doing like lower back area, you don't want to touch the teeth. And this is an example of a friend helping with the cheek swab. So you'll do one on one side, and then you just screw off the top and invert the tube and then shake it. The whole thing comes with directions and kind of picture directions of how to do it as well. And then you can put it back in the prepaid UPS envelope and then either schedule a pickup for the UPS or drop it off at a UPS drop-off box. Or other location. We ask that if you're able to just send us an email that you sent it out just so that we can follow the tracking for it and make sure that we receive it. And then that is once we have a big batch of a whole bunch of the samples collected, that's when we're able to send them out in batches for the sequencing to take place. But once we obtain it, that's how we store it appropriately until it's ready to be shipped out for that type of testing then. But super easy to participate. We're able to do 500 genome sequences, but we're able to really consent anybody into the project and getting their medical records kind of for that review of everything and comparison for as many people as possible. But we're really hoping within the next few months to be able to hit our goal of 500 samples. And then next slide. And we wanted to just make this short just so that we can help answer any questions that you may have. But this is our team and also when the Dravet staff came out to visit us. But a lot of the communication you'll be doing by email are with Angela Winters, who's on the call, and Monani, who's on the lower right. And then, of course, Anna and Megan Kosiska are part of the project as well. And that is how you can email us. But would love to take any questions that you guys have.
Thank you guys so much. Does anybody have any questions? I see one that's come in from the chat asking if you received samples from Mexico, but can you answer broadly about international samples?
Unfortunately, at this time, we're not accepting international samples just because of some of the logistics behind it. That's not to say, I think for this specific project, no, but that's not to say that in the future there won't be other opportunities for that to happen. Oh, there is, so the question is there a minimum age requirement? There is not a minimum age and there is no maximum age either. We would love this to be a very diverse group of samples. So we probably, I mean, for the gain of function SCN1A, we probably have kiddos who were around like 2 months old maybe, but then also individuals who are older and have SCN1A, we would love those samples as well. And we realize that records might be hard to obtain for those older individuals, but we will take whatever records are available.
My daughter's going to be 38, but most of her younger life was all her stuff was at CHOP. So she would be pretty easy to track.
I wonder, I'll have to talk offline with Anna about if we can get into the archives of the paper data too.
Yeah, no, but so the answer is absolutely. Yeah, I think this is especially when it comes to sorry, do you mind if I quickly kind of add a few points there? So, Amingo Helbig, I'm the PI of this project, and the main thing is really that the genome doesn't change, yeah. But I think especially adults with Dravet, they have so much more, let's say, yeah, medical history to them that is important for us to interpret. As you can imagine, if we have a 6 months old with Dravet, we don't actually know what the developmental outcome is. For someone who is 38, like your daughter, we have a good sense on how this person has presented in the different domains that Dravet manifests in. And I think the really unsolved question that we're trying to answer is what causes that? Why are some people with Dravet, why are they more severely affected with their intellectual disability? Why do they have more autism? Why do they have more severe seizures, whereas someone else is seizure free? And I think people have thought for a very, very long time that it's in the SC1A mutation itself. Yeah? With a few exceptions, we know now that this is actually not the case, and we're looking for the what is called modifiers. And we're trying to understand if you have Dravet, what really determines how severely affected you are in these different domains.
Like, Kelly, you have your hand up. Do you want to come off mute? I do see one question that came through in
Oh,
the chat.
sorry.
Oh, go ahead, Kelly.
Sorry. I was having trouble finding my mute button. So I have a question. I actually have I have a daughter that passed away in 2010, and we were involved with the SUDC Foundation. This was before we knew anything about Dravet syndrome, and a couple of years after her passing, we had a subsequent child who was diagnosed with Dravet. And we later were able to go back and do some testing for our daughter and discovered that she also had the mutation. So I guess my question is, would we had the whole exome done? Is that something like, would you be interested in that information as well?
Anna, do you want me to take this?
Sure.
So this is, I mean, an unsolved problem. So I think for your so first of all, I'm very sorry to hear that your daughter passed away. We actually know the SUDC project very well. We actually talked to Oren Divinsky and Laura Gold.
Yes. Okay.
And we know that they may still have biosamples and DNA. Yeah, so exomes. Um, um, we have probably we have done the analysis for them, so we may have even kind of done part of the analysis. What we need for the project is actually the DNA, because we're going to do a different test. We're going to do genome sequencing. So exome sequencing is only partially helpful, but we can definitely connect. Yeah. Do we have your other daughter enrolled in the Dravet Genome Project?
I don't believe that my son is enrolled. No, we
Oh,
are
for your son,
interested
yeah,
in getting him
yeah.
enrolled.
So and I would say I think for your son, this let's say is an easier process, because we've seen
Sure.
just logistically getting information on people who have passed away is actually, it has many challenges. Yeah. So I
Mhm.
think there are some things like the SUDC where we kind of work with them and look at the genomic data almost like in the same way. We're just trying to ask the same questions there. But I think it's often sufficient for us to have a single person per family that we can consent. Yeah.
Okay. Thank you.
I think next is it Koki that you had your hand raised.
Yes, yeah, thank you for the presentation. Um, I think I know the answer to the question from the website, but I still want to ask. So if we send the sample along with we have such and such uh gene mutation, but we don't get the like specific results back from this for our mutation, we end up in this kind of severity, this like the map, like relative mapping or answer or diagnosis or any kind of feedback to this particular gene mutation along the course of this experiments or scientific publication?
So you're asking that you don't your child has Dravet, but you don't know exactly which mutation this is, right?
Oh, we do know, but I think what I think what everybody is probably interested is like how I think from the like larger study of this data that you have kind of the mutation and then the outcome, maybe you can get better like I don't know, like um mapping of where this gene mutation maps in the, I don't know, let's say like severity range or the kind of projection of the, I don't know, what this gene is strongly associated with this type of development over the, I don't know, course of the, like those kind of like individual feedback um for your child.
Yeah, so and this is actually a good point. I think so. There are like maybe three parts involved in this. And maybe Anna, you can just kind of comment on this too. First of all, it's like the SCO&A mutation itself, you know, then other mutations, and then the kind of things with that were so other mutations in other genes that that may be relevant. And then the third thing is basically the results of our specific study. Yeah? And what you mentioned was like individual feedback. I'm going to maybe just walk you through. So typically, we enroll people who have a diagnostic variant in SCN1A. And again, with the exception of a few, let's say, edge cases, like the early onset SCN1A or like milder GEPS plus, the answer on why someone is different, where someone has more intellectual disability or less severe seizure is often not in the SCN1A mutation itself. Now, this is something that we get clinically very often, this question that a Dravet mutation is earlier or later in the protein, and does this actually matter? And it's now actually established that this is not the case. Yeah, so I think this is something that is important to know that if you have a stop mutation, for example, in Dravet, it doesn't matter whether it's in position 10 or 20 or 100. Yeah? When we consent people for our genetic study, first of all, it's a research study where we are kind of asking for your help. Yeah? But there's some ways where we can feedback information, and there's a very, let's say, clear framework of what information counts as what is called actionable. Yeah? For example, if we have a way that if there's, for example, like a cancer mutation in someone's genome and we come across this, we have a way to feed this back to someone. If someone has like a different condition in addition to Dravet, there is a way that we can feed this back. This doesn't happen often because we're not doing a clinical test. So everybody who has a Dravet test has already kind of gone through a clinical genetic test. Yeah? So we're looking at the things that kind of modify Dravet. And those are things we're basically looking at. Yeah, so this is not data where it's immediately clear what we're going to find and what we're going to feedback. We're doing this because this is very early. We're trying to understand what modifies Dravet. And once we understand this, then we'll feed this back. And we don't quite know yet whether this will have the, let's say, quality and strength to really be applicable to any single person. Yeah? But I think those are things that we don't know yet. This is what we do research.
I have a question for you. So my daughter started having seizures when she was 5 months old, and
Yeah.
she's gonna be 38 in a few weeks. But we really didn't know that she had SCN1A until a couple years ago. And she's had brain surgery at CHOP. Would that disqualify her for this? That she had surgeries and
No,
I mean, like,
no.
she's 8 years ago, we almost lost her. She aspirated and I was put in a coma and paralyzed and, on a trach. And somehow she survived. They don't know how, but she survived and she's, knock on wood, she's doing pretty good. So we even have all that documentation too of, what was going on with her 8 years ago, where it came pretty close. But the brain surgery wouldn't disqualify her from this.
No, no, and I think that so our study is a genetic study, and genes don't change. Yeah, so I think this is something where this does not kind of whether you're 3 months or 3 years or 30 years, it doesn't actually change your genes. Yeah. And I think so Pam, first of all, thanks for kind of sharing the story because this is a very typical story that
Yeah, well,
often
I'm
happens.
definitely count me in.
Yeah, no, absolutely. And I think that those are just important because we want to understand why people with Dravet have more severe epilepsies
Yeah.
than not. Yeah. And this is really, think of this really as like an open question where we think there might actually be ways how we can score this or measure this through other things in the genome other than SCN1A. And given that the differences between our Dravet kiddos and adults can be so striking, even though they have the same mutation or same mutation class. Yeah, there is so much to be answered out there that we need to figure out. Yeah?
Well, thank you for doing this. This is wonderful news.
I think we jumped over Vicky. Vicky, you've been very patient with us.
Oh, that's okay. Actually, while I was being patient, I think you basically answered the question that I was going to ask. I'm in Australia. I'm really disappointed that you're not accepting international samples. And I suppose I'm really interested now to know what is the timeline on this before we can start seeing some results? Like, are you
Yeah.
doing this over, is this going to take you? Because I have no idea what each of your individual testing of the samples, how long that's going to take. But I'd just like a sort of around, is this like a study for, 12 months or you don't know? Or,
So I think, and maybe Anna and Sarah, may I ask you to kind of comment on this. So but basically, we have so this is a research study, and I think the reason why we cannot use international sample is just mainly through not so much for consented people, we can consent people from around the world, but actually the process of shipping DNA.
yeah,
But this
yeah,
has to do with kind of like biological material, how this kind of
yeah,
crosses border, and this is something that's actually very, very challenging. Yeah, so this is
yeah.
something that is um, something that would be at this point more difficult than to do. I think there's going to be once this is successful, this will hopefully be like a larger study. Like I know from Ingrid Scheffer, for example, who I've trained with, that the Dravet community in Australia is very, very active. You know, I know that there's lots of things going on in Mexico. There will be follow-up studies. Right now, we're at what were the numbers, Sarah? 470. So we're looking.
77.
Yeah, so we need 70 more individuals, and in this initial part of the consenting will be closed, and we need to analyze this first. This will happen in the next 12, 15 months. Yeah.
And that makes sense what you're saying about the, not taking it from international because, yes, I suppose the degrading of samples and then actually even getting them through across the border may be difficult. It
Yeah.
might all make sense. So
Yeah,
thank you.
and this will at some point happen, but at this point, this is not the, let's say, the best way to answer this question. Yeah,
Okay.
and I think so. First of all, thank you for being interested in this, and I always hate to disappoint people, but in this case, it's basically um, in a way necessary to given how the restrictions are about genetic data. It's meant to protect people. Yeah.
Yeah, that makes sense. Yeah. Thank you.
I think if we go to the big question that everyone's asking for timeline, when do we plan to release information of individual kids' genomes? We will not be releasing that information. The testing, feel free to interrupt me, Dr. Helbig. The type of genome that we're sending is done on a research basis. The way that the study's set up is to be on a research basis. It would require us to have a different protocol to be able to do something more on like a clinical basis. There is, Dr. Helbig said earlier, if something was clinically actionable, you can opt in or out to be notified of that. But that would be like another kind of monogenic thing that we would find. It wouldn't necessarily be related to finding these like multi factored type of genetic underpinnings that we're trying to look for. But this project's really is based on like the broad trends that we're seeing and putting this big data together to put that out. So you might notice your child's gene variant in some of the research, but it would be in the context of a bigger setting for the data.
Yeah, and I
Hopefully
mean,
explain
so
that
basically,
okay.
this is kind of done under our research studies at Children's Hospital Philadelphia, which is one of like the strongest protections for privacy and health information that you can imagine. Yeah, we're not trying to do something trivial. So this is not genetic testing that you can kind of get at your geneticist or kind of order clinically. We're trying to answer new questions, and this is not anything that is, let's say, built yet into workflows. We're trying to do this. That's the goal, that we can say we can find a score for someone. We can just kind of develop a score that we can tell someone at the age of 6 months, this child is more likely to have significant more impairments developmentally. So we have to just kind of really be on our toes very, very early. Or this child will likely have a milder epilepsy course. Yeah, just imagine what this information could help us with. Yeah. And this is done in other fields of medicine. This is done in cancer. This is done in other conditions, and we want to get to Dravet there. This is what we need your help for. Yeah. So this is not a test where there's an easy answer. There's going to be lots of experimentation, how we analyze this data, lots of things we have to try out. Yeah, but we can only do this if we have people helping us with this. Yeah.
At any point, would you want to have see any of these children and young adults at CHOP? We live pretty close.
Yeah, so I think we at this point our kind of request is very modest. At this point, we just need samples. Yeah, we've kind of built an entire workflow on how we can ship these kids to samples. We don't actually need people to come in, which is nice.
Okay.
Yeah. And then I saw the question in the chat. Yes, this is also for SC1A gain of function. We kind of made a case that we want to include them too. Yeah. My personal opinion is they belong into the Dravet Syndrome Foundation. Yeah. This is, I think, Veronica and this is an ongoing discussion, but we want to include them too, given that this is, still, let's say, a largely unexplored group that we also feel responsible for.
Absolutely. Koki, did you have another question?
Yes. Is there any like a detailed research proposal or something that concretely describes what you are planning to do as experiments? Like, for example, some research might collect the, like, say, onset of the seizure stars, and then maybe gene mutation, and then maybe use such and such machine learning algorithm to predict the outcome, or like that level of details. I saw on the website you say something like outcome, like seizure severity, development, risk of SUDEP. But is there any like proposal or something that describes what kind of input or like analysis you run, and then what kind of output or method that you use along the course of this experiment?
So, Koki, this is actually a very good question. I think this is kind of, and I hear that you have some background there in research, and maybe I want to broaden this out to everybody. So, research is something that we kind of ask families to help us with. Right? So this is the contrast. So Sarah, Anna, and I, we work on our engine, our epilepsy genetics program, where we kind of do clinical testing, where we see our Dravet kids clinically. So much of what we do is for families. When we think about research, this is something where we ask families to help us. Yeah. When it comes to how we consent families for research, there's actually, we try to leave this actually relatively open on what type of analysis we do. Because we don't actually know if this changes. So this is the difference between you come into our clinic and we say we have to just kind of send off a blood test to check a child's step code level, or we have to just do an EEG just to understand how frequent are the discharges and are these twitches we're seeing at night, are they seizures or are they not seizures? When it comes to research, especially to kind of genetic research, we basically ask families to give us permission to use the data broadly. Yeah, broadly means not carelessly. Broadly means on a very, very clear definitions of how we can use this in information. Yeah, but we're not actually just kind of defining exactly what we're going to use this for. Yeah, there's very much development right now in how people understand genome data, how they understand clinical data. Yeah, Anna is now putting together actually one of the first clinical papers from the Dravet genome study, the first batch of 100 samples, and this is evolving. Yeah, so when we try to be a little vague and say we're not specifically saying this is the type of analysis we're doing, this is on purpose to kind of allow the field to advance. Yeah, and eventually this is meant to be a controlled access data resource, something that other people who are qualified and who are vetted can also access. Yeah, so we have to keep this broad in order to allow all questions to be answered.
Okay, sounds good. Yeah, thank you. So it sounds like it's more open-ended and kind of more future-proof in case there's a better or interesting research
That's exactly.
comes
So
out.
that's language. Perfect. That's language that sometimes use. If we want to future-proof this, because it has been actually a very sad situation in the past that some research studies and some hospitals have built biobanks and biosamples that were very narrow. Yeah, where people couldn't do actually the necessary analysis and like understanding stalled because they could only, they could analyze X but not Y. Yeah. And this is something where we're trying to just really kind of create a resource where we can have families permissions, where it's very clear that we're never going to share names, we're never going to share any identifying information. Everyone handling this data is part of a vetted research team, by NIH, by CHOP criteria, and these rules are very, very strict. But how exactly we analyze the data and what type of analysis we're doing, this may change tomorrow, right? This is research. Tomorrow there may be a big study come out and say these are effective treatments for Dravet, those are kind of predictors for Dravet, and then we need to be able in the situation to look at this.
Yeah, thank you. I really like the way of thinking.
Thank you.
I'm not seeing any more new questions in the chat. Does anybody else on the call have a question that didn't get answered?
And maybe while we're just waiting for questions, I think one thing to say, this is really a joint project between DSF and our center. I think this is one of these studies where we, Veronica, we just kind of stood this up together. There's lots of discussion being back and forth how we would like to do this. And I think this is something where the Dravet community can already be proud of. No other condition has done this yet. This is the first genetic epilepsy where we're able to kind of get these numbers and really ask these questions and build these resources. And we just need to get this across the finish line. We need your help to get all the remaining samples in. And this is going to be,
I was
paradigm.
I was curious
Yeah.
if having a sample from my dad would be helpful to you. I guess he has Jeff's Plus, and I myself have Jeff's Plus, and then the epilepsy has just gotten progressively worse with each generation. And then my son has Dravet syndrome, and he has a double variant SCN1A mutation, but he's the first one with Dravet syndrome. But he's the sixth generation with epilepsy. But the first one with Dravet syndrome.
Yeah. So Jennifer, I always want to be a little bit kind of careful that I don't talk about individual stories too much, because this is often we're talking about in the public forum. But I would say in this case, your son and you, just for
I have epilepsy, and my dad has epilepsy.
Yeah, and I think we want to we typically kind of think about the most severe person in the family. If there are double mutations, then I would say we've got two people in the family, and I would definitely suggest that this is of interest for you to be part of the Dravet Genome Project.
Yeah. But his genetics, when he got genetics done at age 3, said he got a de novo from his father, who does not have epilepsy. Showed the SCN1A de novo, and then the disease causing from me SCN1A mutation. So he got the two variants, I guess.
Yeah, no, I think this is something I think once you talk to our team, maybe we can just dive into this a little deeper. And maybe this is something that you can just kind of, if you can send Jennifer to maybe you can just look into this, yeah. But I think the idea is all these are so many Dravet stories are uncommon, right? This is something, there are lots of things that are common, but you kind of notice that every child has a unique feature, yeah, and
Yeah.
those things are like a unique twist to their epilepsy, to their history. And what we're trying to say, kind of, do we see some patterns in there that we can help explain genetically beyond SCN1A? Yeah, and the reason why we're just kind of so insistent on this is because there is a lot. There's a lot of genetics outside of SCN1A that kind of, yeah, determines how we do. For example, the genes that cause, yeah, height, why some person is kind of taller than the other, why one person kind of, yeah, has is intellectually more advanced than someone else. All these things are probably magnified in our Dravet kits, and this will be very important if we have this as, yeah, if we have an understanding to move this forward. Yeah.
I didn't know if the double variant makes it less severe or
So Jennifer, I'll actually maybe I'll have Anna reach out to you maybe to kind of clarify this offline. Yeah, but I think this is an important question. Yeah.
okay.
We've been in touch by email, so
Yeah.
if you have any more questions, just let me know.
Yeah.
Okay.
I saw a couple of questions in the chat from families that have been patients at CHOP in the past that were assuming that because they had their genetic testing done, they were automatically enrolled. So I just want to confirm that that's not the case and that they would need to submit the cheek swab and consent for this particular study. Is that correct?
Yes.
Absolutely.
And this is on purpose. So I think when you think about it, it's like hospitals don't automatically
Mhm.
do, yeah, stuff with your samples that you don't know. So basically, the idea is that even within CHOP, we're a research team, but that no one else in a way has access to this. This is meant to be like data protection, yeah, and this is meant to kind of, yeah, keep people's privacy safe. So if you have been a patient at CHOP, unless you're enrolled in the Epilepsy Genetics Research Project, yeah, don't assume that we have your data. If you have not talked to our team, don't assume that you're part of this. This is on purpose, yeah, because this is meant to protect privacy. We can do de-identified research with some data, but not with genetic data. This all needs to be a one-on-one discussion. So if no one has approached you about this, please approach us. Yeah.
And I also wanted to just comment on people asking about kind of the timeline for this project and for our results. So we are committed to kind of doing things and giving this information and what we get from it back to the community as fast as possible, which is why we're kind of working right now on an interim publication focused on kind of the clinical features of the patients or many of the patients that we have enrolled so far. But in order to kind of do what really is the point of this project, which is to combine that clinical information with genetic information, we need to have the power of having all 500 people and we need to have their genomes done. So right now we're really focused on getting those 77 last samples so that we can, as quickly as possible, send them for genome sequencing. And then after that, we can begin working on the data analysis. So I think that the timeline really depends on us getting those last samples, which is why we're, here today and asking you all to participate and really do appreciate all of your enthusiasm as well.
All right. If anyone has follow-up questions that you don't feel like I answered today, you can always reach out to SCN1A at chop.edu. Is that correct? Okay. And then also you can reach out to DSF as well. I'm just Veronica at dravetfoundation.org. So if you have a research related question that isn't just about this project, or you forgot their email and you need to be connected to it again, you can always reach out to DSF and we'll help you connect to who you need to. I think
Kobe,
we have one more question.
Kobe, we don't have to stop. We take more than 500. Yeah, but it's and it's like there's always like samples that fail and things that have to be redone. So this is an ongoing recruitment, but eventually we kind of eventually have to see how we continue this. But right now we're continuing the recruitment. Yeah. So please help us. Yeah, so this is something for all Dravet families. We're really trying to look at some aspects of Dravet that are within reach. Yeah, and this is the first genetic epilepsy where we're trying to look at these kind of other things in the genome. This has to be done at a certain scale. This is not just done in a single person, but we need to understand patterns there. These patterns are out there and we'll find them. Yeah, but we need your help.
Thank you so much, Ingo and Anna and Sarah and Angela, for joining us today. Thank you to all the caregivers and families that joined the call today and asked, I think, an excellent set of questions that helped clarify this, I think, for everybody. If you've already participated in this study, thank you so much. If you're considering participating, thank you so much. It's been incredible to watch the community ask these questions, which inspired researchers to develop these research plans to answer them, and then to see everyone make it happen together has been really amazing to watch play out. And I think seeing all this data come together, hopefully in the future, is going to get us some insights and hopefully exciting answers and maybe new questions to look into more. So thank you to everyone.
Thank you.
Thank you.